Hello everyone. For my seminar topic, I will be discussing nucleotide degradation and chemotherapy. And for the first 2/3 of the video, I will cover the catabolism of purines and pyrimidines, and then I'll finish the video off with chemotherapy and chemotherapeutic agents. So for purine degradation, we will observe how adenosine monophosphate, also known as AMP, and guanosine monophosphate, also known as GMP, are broken down to form uric acid. So the first step in our catabolic pathway for AMP, we are going to remove the phosphate group in the 5th position using this enzyme called 5' nucleotidase, and that's going to be assisted with water to pop that phosphate group off. And then next we are left with adenosine, which is just our purine base adenine, and then our sugar ribose. And then this enzyme adenosine deaminase is going to come in and remove this amine group, which is also hydrolyzed, to relieve us with our amine leaving group. And basically this enzyme comes in, removes the adenine group, and then we are left with the carbonyl. And so now we have inosine, and so to remove the sugar, we're going to use this enzyme called nucleosidase. And this is also hydrolyzed to relieve us of ribose. And so now we have hypoxanthine, and that's going to be oxidized with xanthine oxidase that is coupled with the reduction of oxygen to get us xanthine. And then that is further oxidized again using the same enzyme. And reduction to get us uric acid. So next we are going to go over the catabolism for GMP, and it follows the same pathway as AMP. It's just a slight difference based on the structure of the nucleotide. But initially, we're going to use the same enzyme, 5' nucleotidase, that is going to come in and remove our phosphate group. And then we're left with guanosine. And since our nucleotide already has our carbonyl in the right position, there's no need to deaminate anything yet. So we're going to remove our ribose sugar using nucleosidase and water to relieve ribose. And that's going to leave us with guanine. And now we're going to deaminase to remove this adenine group and replace it with a hydroxyl group, as we see in xanthine. And that's also hydrolyzed, and our amine group is relieved and then replaced with a hydroxyl group. Then xanthine also is oxidized with the couple reaction of reducing oxygen, and then we are left with uric acid. So for the overall pathway for degrading purines are going to have the same enzymes depending on the nucleotide that you start with. So if you're going to use GMP, you're going to remove the sugar first using nucleosidase and then deaminate. And in AMP, you're going to deaminate first and then remove the sugar because of the structure of both of these nucleotides. But the end product is going to be the same. We're going to get xanthine, and then we're going to oxidize, and we're going to end up with uric acid. So we just learned how our body is able to produce uric acid by processing purines. And in most animals and mammals, we are able to further degrade uric acid to produce allantoin using urate oxidase. In other organisms, such as fishes or amphibians, they are able to excrete more nitrogen by metabolizing allantoin into ammonium. And the enzymes that are involved are allantoinase to produce allantonate, and then allantonate is broken down by allantoinase, and that produces urea, and urea is broken down using the enzyme urase to produce ammonium. And you can find this breakdown in our book, or the 8th edition, figure 2248, and it just gives you a flow chart of the breakdown of uric acid or degradation of uric acid. Before I go on to the catabolism for pyrimidines, I would like to mention how purine degradation can also synthesize more nucleotides via salvage pathways. And how salvage pathways work is by taking free purine bases that are available during the catabolism of purines and recycling them to make more nucleotides. So adenine and guanine can be recycled to make more AMP and more GMP. Another side product known as hypothalane can be used to make IMP, also known as inosinate. So the salvage pathway is an alternative way to synthesize nucleotides besides our biological pathway known as the de novo pathway. And this also helps with some regulation, and because our body is not going to be breaking these recycled bases down into uric acid, they're going to be used to produce more nucleotides, which I guess gives our body more time to process uric acid, and that way it reduces the production of uric acid. So yeah. So now we are going to go over the degradation of pyrimidines. The two nucleotides that we will observe will be thymidine monophosphate and uridine monophosphate, also known as TMP and UMP. So before these nucleotides are further degraded, they go through similar, or they use the similar enzymes for purine degradation to isolate or free the purine bases, as we see here, thymine and uracil. And they use the same exact enzymes, nucleotidase or nucleosidase and dimease. I know it's hard to see, but we've been through these enzymes when we went over purine degradation. And those enzymes basically just get rid of the phosphate group and the ribose sugar to leave you with your purine base. And then I will go over how these purine bases are further catabolized in their own separate pathway. And thymine produces methylmalonyl semialdehyde, and uracil produces malonate semialdehyde. So starting with thymine, our nucleotide is going to be reduced using dihydrouracil dehydrogenase. That is going to be coupled with the oxidation of NADPH to NADP plus, and that's going to form dihydrothymine, and that is going to be hydrolyzed in the presence of dihydropyrimidase or dihydrothaminase, depending on the pyrimidine that's being used. So if we were using uracil, this enzyme could be referred to as dihydrouracil, and that is going to be hydrolyzed to form beta-uridoisobutyrate. Respectively, this enzyme beta-uridopropionase is going to remove an ammonium ion and a bicarbonate in the presence of water. So we're going to get ammonium ion and bicarbonate, and that is going to form beta-aminoisobutyrate. And last we have aminotransferase, and that is going to be coupled with the transamination of alpha ketoglutarate to glutamate. And that is going to form our methylmalonyl semialdehyde. Okay, so now we're going to go over the catabolism for uracil. And uracil goes through the same catabolic pathway as thymine. The only differences that we're going to see is going to be in the intermediates and the name of the enzymes, because the structure of uracil does not have that extra methyl group like thymine does, but everything else is going to be pretty much the same. So uracil is going to be reduced using dihydrouracil dehydrogenase coupled with the oxidation of NADPH, and we're going to form dihydrouracil, and that is going to be hydrolyzed in the presence of dihydropyrimidase or dihydrouracilase. I think, I don't know if that's correct, but it doesn't really sound right, so I'm just going to say dihydrouracil hydrolase. And then that is going to form our beta-uridoisopropionate, and like I said before, it's no longer butyrate because uracil does not have that extra substituent or methyl group. So we are moving from 4 carbons to 3 carbons, so we have that propionate. And then we're still going to be using this enzyme, beta-uridopropionase, that is going to take off this ammonium and bicarbonate group in the presence of water and then we're going to form beta alanine. And once again, aminotransferase coupled with the transamination of alpha glutarate is going to give us our product, malonate semialdehyde. And we've all seen this reaction before with alanine and the degradation of amino acids to form ammonium, which was the precursor for the urea cycle. So in purine catabolism, we learned that purines can be degraded into uric acid and then further processed into allantoin. But in pyrimidines, they are often catabolized to form ammonium or used for the urea production. For example, in thymine catabolism, we just learned that that is degraded into methylmalonyl semialdehyde, which is an intermediate in the valine catabolism. In valine catabolism, methylmalonyl semialdehyde leads to the production of succinyl-CoA, and then that can be converted to fumarate and then malate and then oxaloacetate, and then that can be changed into arginosuccinate, which is a direct intermediate in the urea cycle. So another thing that is similar to the degradation of purines is the Savage pathways. And just like we saw in pyrimidines, pyrimidine degradation can use the free bases of the nucleotides to recycle and make more nucleotides instead of further producing this to form ammonium or urea production. And this is done via the enzyme called phosphorybosylase transferase. Which is an alternative way to synthesize these nucleotides. Now I will briefly discuss the importance of understanding nucleotide metabolism and how that knowledge contributes to the treatment or chemotherapy for cancer patients or any other patient suffering from a disease. So in most cases, the acceleration of this disease is diagnosed by uncontrolled cell growth that mitotically cluster to form these tumors, as we see here. And our biological regulation to manage excess cell growth is no longer inadequate, meaning apoptosis can no longer process cell death at a sufficient rate to maintain cell division. So this is no longer adequate or sufficient enough. So the next step in managing this rapid cell growth would be by regulating the DNA replication. And we know that DNA is composed and expressed by its nucleotides. And since we are biochemically familiar with the metabolism of nucleotides, we can control the replication of cancer cells by interfering with the synthesis of nucleotides. So we interfere with the synthesis of nucleotides, we can reduce the replication of cancer cells. And this is where our chemotherapeutic agents come in. And two therapeutic chemotherapeutic agents that are used are deoxynucleoside monophosphate, also known as FDUNP, and then also methotrexate aminopeptrin, or MAT. And what these do is they interfere or inhibit the enzymes involved in synthesizing nucleotides such as DTMP. So by stopping the production of DTMP, which I will explain in more emphasis in a little while, cancer cells cannot replicate their DNA because they cannot complete the genetic code that's required without DTMP. And that's why FDUMP and MAT are good chemotherapeutic agents. Lastly, I just want to elaborate on why FDUMP and MAT are good chemotherapeutic agents. So fluorouracil is recycled in our bodies via the salvage pathways and then converted to FDUMP. And then that interferes with the enzyme called thymidylate synthase, which assists in the conversion of DUMP to DTMP. And then we also have MAT that comes in and interferes with this enzyme known as dihydrofolate reductase. And that also assists in the conversion of DUMP to DTMP. So with both of these chemotherapeutic agents involved, the synthesis of DTMP is interfered, and therefore DNA replication can no longer occur. And without DNA replication, the cell can no longer grow or divide. So I wanted to add one more thing and explain why these chemotherapeutic agents stop the synthesis of DTMP and not DUMP. And it has a lot to do with our RNA and DNA. And we know that our DNA is made up of DTMP and our RNA is made up of DUMP. And our RNA is very essential to our biological process, and it's responsible for the transcription and translation and protein synthesis. So we do not want these chemotherapeutic agents interfering with our RNA synthesis. And we could afford to temporarily stop DNA replication because we can always make more using our RNA template. And although DNA is responsible for a lot of our active cells, such as our hair, our nails, and skin, things that constantly shed and grow daily, we want to attack DNA because it also involves a lot of our new and foreign DNA, such as cancer. And so the best way to do that by controlling cell growth or controlling foreign DNA without harming our own biological RNA is by stopping DNA replication. And yes, we get a lot of side effects by doing chemotherapy because it interferes with these active cells. And that's why people vomit, or get pale or lose their hair, because there is no more DNA replication occurring for those active cells. However, it's very good for preventing our cell growth and cell division for new foreign DNA, which we want for cancer or any other disease. And so that's why those chemotherapeutic agents are preferred, because we protect our biological process, but at the same time we slowly eliminate our foreign or new DNA. Just to conclude what we went over, we talked about nucleotide degradation in purines and pyrimidines. We also talked about their savage pathways. And then we ended with chemotherapy and chemotherapeutic agents such as FDUMP and MAT. Or chemotherapy for case Why can I say this shit? Now I will briefly